Let's in vitro. Now the technology used is known as hybridoma technology. Now antibodies can be of two types: polyclonal antibody and monoclonal antibodies. Now these polyclonal antibodies are produced during this microbial infections. And what do you understand by polyclonal? All different types of immunoglobulins are produced whenever a pathogen attacks a human being. Now what are these monoclonal antibodies? These monoclonal antibodies are mainly produced in multiple myeloma. Multiple myeloma is a malignant transformation of a single clone of plasma cell, and there can be production of only IgG or only IgM, especially the light chains. Now, so it is mainly characterized by production of specific single immunoglobulin. So this is whenever a individual develops conditions like multiple myeloma. Other than this, monoclonal antibodies can be produced in the laboratory by hybridoma technology. Now the advantage of this monoclonal antibodies is these antibodies are specific against a particular antigens, and they are required in less quantity when compared to polyclonal antibodies for immune defense system. Now, apart from that, where there are a variety of applications of this monoclonal antibodies. These are employed in enzyme-linked immunosorbent assay to identify any antigen or protein molecule which is present in very less quantity. Apart from that, nephelometric assay where light scattering property is utilized to estimate or quantify an analyte, biochemical analyte. Apart from that, monoclonal antibodies are used in cancer chemotherapy so that the only cancer cells are destroyed and the host cells are spared. So monoclonal antibodies are also used as immunosuppressant for graft rejection, to prevent graft rejection they are used. And the variety of application of monoclonal antibodies lies in estimation or quantification of enzymes, drugs, hormones, viral antigens, which are usually present in micro quantities in blood circulation. So these are the application of monoclonal antibodies. Coming to the technique of hybridoma technology producing monoclonal antibodies. So what happens is normally normal healthy cells from mice are picked up, and these are usually the T cells, and they possess these following characteristics. So they are HEPRTase positive. HEPRTase is the enzyme from salvage pathways of purine, so that denosynthesis of purine can takes place. And these are HAT resistant, and HAT stands for hypoxanthine aminopterin and thymidine. So they are resistant when they are added to this particular media. So these normal T cells have got the capacity to synthesize and secrete immunoglobulins, but they cannot proliferate. So these are the characteristic feature of normal T cells that are isolated from mice. So the myeloma cells that are isolated from mice possess these particular characteristic features. They include HEPRTase negative. They do not have this enzyme, so they cannot the myeloma cells cannot synthesize purines and pyrimidines. They are HAT sensitive. That means if these myeloma cells are added to culture media containing hypoxanthine, aminopterin and thymidine, so they tend to die. They cannot survive. So they cannot synthesize immunoglobulins, but they have the capacity to proliferate. So the only plus point of myeloma cells is ability to proliferate, and whereas the plus point of the helper T cells from the mice include they can secrete immunoglobulins, they are HAT resistant, and they possess these enzymes for synthesis of purines. So now these two cells, the normal T cells of the mice are fused with the myeloma cells of the mice using polyethylene glycol, and after fusion they are added to this HAT medium, which contains hypoxanthine, aminopterin and thymidine. So what will be the result? We'll have unfused lymphocytes, and they die because they do not have the capacity to proliferate, and unfused myeloma cells in the culture media also die because they do not have this enzyme, HEPRTase enzyme, so there is no DNA synthesis. Only fused cell will be able to grow in the HAT medium, and these few cells are known as hybrid cells. Why these hybrid cells have got the capacity to grow in HAT medium? Because now these few cells have genes of both the normal and the myeloma cells. So they possess this HAPRTase activity of the normal cells, and they utilize hypoxanthine and thymidine, which is present in the HAT medium, for DNA synthesis, and they are able to synthesize immunoglobulins. And further, these cells are HAT resistant, that is they do not die when they are exposed to this antibiotic, that is aminopterin. And due to the presence of this myeloma cell genes, these hybrid cells have capacity to proliferate. So these hybrid cells are isolated and propagated both in vivo and in vitro to produce monoclonal antibodies. This is how monoclonal antibodies are produced, and they are used for therapeutic purpose, cancer chemotherapy, and for estimation of analytes. They used in other technique like ELISA metric assay. So this was all about hybridoma technology. Happy learning. Stay tuned for next videos.